Hello, I'm Momna Hejmadi and I teach in the Department of Biology and Biochemistry here at the University of Bath. In this video, I'll talk a little bit about cancer and the complexities involved in designing drugs for the treatment of cancer.
Cancer occurs in 1 in every 2 individuals. However, the news is not so bad because 4 out of 10 cancers are preventable. A lot of this depends on our diet, our lifestyle, for instance, lack of exercise, or diets when low fiber increases the risk of certain types of colon cancers, exposure to the sun, smoking, pollutants. These increase the risk of developing certain types of cancers. But the biggest risk of all is age. The longer we live, the likelihood of developing certain types of tumors increases as we go older. So now let's look at what makes a cancer. How do cancers develop? Before we go into that, it's important to understand how cells behave because a lot of the changes that happen is dictated by cell behavior. And you might not know this, but every cell has a predestined fate. Where it lives, where it survives, how it differentiates, how it divides, all of this is controlled by signals that drive it internal as well as external that drive its progression. And cancer is no different. So again, regulation is key. So if you don't have this regulation, you get chaos. And that is what happens in cancers. And this chaos is defined as genomic instability. And DNA mutations are a key part of this genomic instability. I've shown this in this figure here where you see this clonal somatic changes and the mutations that occur as cells divide. So for instance, you might have a normal cell dividing into 2 cells. One of them develops a mutation. This cell, when it further divides, it carries this mutation and at some stage might develop a second mutation. And so you end up with a subpopulation of cells that have multiple mutations within the same tissue. But you shouldn't forget that this is a bit Darwinian in the sense that this is survival of the nastiest, just like you have Darwin's survival of the fittest. In this case, not all the mutations drive cell proliferation. Only mutations that give that selective advantage to the cell in terms of cell division are the ones that are carried through. And this is demonstrated in this animation where you see you'll have multiple mutations, some of which might allow proliferation, some of it might allow mobility, so it breaks through the basal lamina of the tissue and enters the bloodstream or the lymphatics, and these cancer cells then migrate to different sites, which is the process of malignancy. Now this timing of when these mutations occur and where do these mutations occur. If you think of cells, you have the adult somatic cells, but you also have cells that are capable of self-renewal, the so-called stem cells. And what we know in the past few decades now, that there is a whole subpopulation of mutations that can occur long before even tumor initiation happens. So this change, mutations occur in stem cells, and these mutations are carried forward, and that's what you see as cancer stem cells. And this is very commonly demonstrated in certain types of lymphomas where you have, let's say, a normal tissue and it carries stages of differentiation. You might have one mutation that develops or multiple mutations that develop that is carried on in subsequent dividing stem cells. And as a result, later on, at some later point where you have differentiation being affected, and that results in abnormal cells being created. When it comes to thinking about how can we tackle cancer, the goal of any type of treatment is 3 things. First of all, the dosing. It has to be at the right dose to reach efficacy, to damage the cells. How long the drug should be given for, the duration, and of course, the third and most important is trying to manage any off-target effects or adverse side effects from the treatment. So the optimum efficacy is a balance of all three that hits the spot just the way you want to target, only selectively the cancer cells and leave the normal tissues intact. And this is achieved historically over 100 years or more through these main pillars of treatment, which is, of course, for most solid tumors is surgery, taking the tumor out. But you also have radiation and chemotherapy, which essentially a bit like using a sledgehammer to crack a nut. You're blasting all the cells in the hope that most of the cancer cells die preferentially, and you have as little damage as possible with the normal tissues. Adjuvant therapy are therapies that combine, let's say, either radiation or chemotherapy with something else. And I'll give you an example of an adjuvant therapy that I've researched on. My early days was a new type of drug called as a bioreductive. And how does that happen? So just let me give you a preview of, let's say you have a solid tumor. The solid tumor is growing bigger and bigger. But as it gets bigger, it gets further and further away from the blood vessels and its access to oxygen and nutrients. And what that means is that you have cancer cells that are hypoxic or have low oxygen environments. And these hypoxic cells are dangerous because not only do they resist any effects of radiation or chemotherapy, but also they actively trigger a type of growth of blood vessels called as angiogenesis. And this combination of hypoxic cells is lethal. One of the reasons why you get relapse in certain types of solid tumors is because of these hypoxic cells, which start to populate the tumor once you've finished a bout of radiation or chemotherapy. And what you see here is this image showing a rat mammary carcinoma. And we stained it with various dyes, and you can see the stain in red is for the endothelial cells, which are the cells that line the blood vessel. Staining in blue are the oxygenated cells. As you can see, these lie very close to the blood vessel because they are well oxygenated. However, as the tumor grows bigger and bigger, cells are pushed further and further away from the blood vessel, and these are the hypoxic cells shown in green. So what happens is when you give radiation or chemotherapy, you're essentially killing off the oxic cells and leaving the hypoxic cells alone because they're resistant to this. It doesn't touch it. And so when you finish radiation or chemotherapy, these hypoxic cells stay alive and they start to divide and you get relapse. So here's a clever trick. You can use drugs called as bioreductives. Now these are typically harmless prodrugs. However, when these drugs encounter a hypoxic environment, they are converted into a toxic compound. And that was where my research came in. I was looking at an alkyl amino anthraquinone N oxide, or AQ4N for short. And that AQ4N is a bioreductive. So when you give it to a patient, what AQ4N does is if it sees or encounters a hypoxic cell, it enters it and it gets converted to AQ4, and AQ4 is toxic to the cell. So here you can get a double whammy. Radiation or chemotherapy will kill off the oxygenated cells, and bioreductives will kill off the hypoxic cells. So this way you get the maximum efficacy by combining these two drugs. If you want to have a look, the paper is on that in the slide. So while radiation, chemotherapy, and adjuvant therapies are well and good, they are efficacious and they're still one of the pillars of standard cancer treatments, they have problems. Side effects is a big problem where you have off-target toxicity caused by this blanket approach to trying to cells, which also damages normal tissues. There's a clever way of going about this, and this is the targeted approach. If we understand cancer, if we understand the genetics behind it, if we understand the mutations behind it, you can target specifically those mutations much more effectively, and it's a smarter way of doing things. And that is also called as precision medicine. In fact, it can even be tailored to individual patients for specific types. Even with breast cancer, for instance, if you have 4 patients, each of them having a slightly different profile, then different drugs can be used by those patients. This is where the complexity comes in. So you might think, that's it, we know what genes there are. Why not just look at all the genes in the cancer cells? And this is precisely what sequencing allows us to do. In the past 15, 20 years or so, sequencing has really made it possible to identify every single gene in any cell or tissue, including cancer cells. And that was where the Cancer Genome Atlas program began. And we now have a better measure of not only what the normal genes are, but also what the mutations are in most cancer types. So the question arises as to how many genes there might be that might be altered in a typical human tumor. And this is where the conundrum arises, or the puzzle arises. It turns out that you can have in cancer cells a very small number of high frequency mutations, mutations that occur at a high level across multiple tumor types. And you also have a small number or a larger number of mutations that occur infrequently. So these are the so-called analogies of mountains and hills. So these mountains are these genes that occur with a high frequency, and the others with a low frequency, but a lot of them. And this is where the problem arises as to how do we know which genes should we target. And this is some clever bit of research that was done by Tomasetti and Vogelstein. Way back in 2013, they published it in Science, where they identified that there are certain genes that are key to driving change or this transformation in cancers. What you see in this image here is different types of tumors, solid tumors and exposure to mutagens, liquids and pediatric tumors. And what you see in the x-axis here is non-synonymous mutations. Non-synonymous are mutations that affect the protein sequence. Yeah? And you can clearly see lung cancers, non-small cell or small cell lung carcinomas, a huge number of mutations and a huge diversity of mutation types too. So you have multiple mutations. On the other end of the spectrum, you have things like hepatocellular carcinoma or glioblastoma or prostate cancer, which have a slightly lower frequency. But if you go further down, if you look at pediatric tumors in glioblastoma or neuroblastoma, the frequency drops even further. And what you see in this picture is sort of the list or the number of mutations that have been picked up on various different tumor types. And you can see everything from 14 to 66, 147 for lung cancers. So certain tissues are more prone to developing mutations. Glioblastoma multiforme is highly aggressive, whereas you might have some sort of hepatocellular carcinomas or liver carcinomas, which are very slow growing. So in order to understand this, what Tomasetti and Vogelstein showed was that there are key driver mutations and these passenger mutations. So if we call driver mutations are the mountains and the passenger mutations are these hills. And we can use this information to design drugs that target this. And one very classic example of this is melanoma, metastatic melanoma. And in almost 70% of patients with this specific type of metastatic melanoma, they have a mutation in a gene called BRAF or BRAF. And this has been identified as an oncogene, and quite a few even common melanomas show this mutation, 50% of those. So what BRAF does in a normal cell, it's a key signaling molecule that tells a cell whether it should divide or not. So it's one of the key pathways involved here. So if you have a mutation in this BRAF, what you have is a disruption of this pathway, and this cell continues to divide. So it's deregulated, and its controls on or brakes on cell proliferation are removed. So if we know that this BRAF is one key signaling molecule involved, and you design a drug against this to stop or prevent this BRAF mutation from working, you block this pathway and you automatically push down cell proliferation. And that is exactly what this drug vemurafenib does. It targets BRAF. It's an inhibitor of BRAF. And let me show you some data from this. Again, for metastatic melanoma, you can clearly see this is the patient's sort of oxygen glucose uptake in the DEXA scan of this patient. And of course, in the brain, you would expect to see a lot of bright spots. So the more brighter yellow-red, that means higher uptake of glucose. And you can see that there's a huge number of tumors in this patient. And 2 weeks with vemurafenib, there's a huge reduction in the number of tumors. And this image here shows, this is really remarkable. It's not very common and not very often that we come across drugs that work at this level of efficacy. And you can see in this patient, about 15 weeks after treatment with vemurafenib, the tumors are all gone. The only problem was 23 weeks later, the tumors returned. And what that means is that there's resistance developing to vemurafenib, and the tumor has evolved mechanisms in which it can bypass BRAF completely and uses an alternative signal to drive proliferation instead. So these are some of the problems even with the targeted approach, even if you know one specific driver mutation that can be used as a target. The promise is some of the immunotherapies that are emerging, and some of the really remarkable successes that we see with immunotherapies, especially with certain types of melanomas, certain types of breast cancers, is these so-called checkpoint inhibitors. And there's a whole host of other immunotherapies. And the principle behind this is that instead of trying to target only a mutation in the cancer cell, why not exploit the immune system of the body and get the immune system to attack only the cancer cells? And this works in several ways, either by stimulation or enhancing the activity of these our own immune system, suppressing or desensitizing certain other types of our immune system, which is normally involved in inflammation. So this is really the promise of the future, where there are remarkable successes with certain types of cancer immunotherapies. So you might ask, okay, that's great. Why haven't we cured cancer yet? And this is where the complexity comes in, because every cancer is different. The mutation profiles that you get, even in the same patient with a primary tumor and the secondary tumor, the mutation profiles might be hugely different. And so drugs that might work with one primary tumor might not be just as effective as the secondary tumor in the same patient. And cancer cells, by their very nature, evolve over time and very quickly. And lastly, it's not just about the cancer cells, it's about the environment they're in. And this is the big thing about understanding the tumor microenvironment, looking at it not from a reductionist point of view, but from a holistic point of view, to see what is the interactions that are going on, what types of cells do you see in a tumor. So this shows you the cancer stem cells, you have inflammatory cells, immune cells, some of which are pro-inflammation, they get rid of tumor cells. Other immune cells are recruited or used by cancer cells to escape. So, these immune cells can't detect the cancer cells, and so they avoid inflammatory attack. There's also, of course, the endothelial cells, other different types of cells that you get in a tumor microenvironment. And the complexity comes with this interaction with all these components. And this is the big challenge in understanding tumors, understanding cancer. That'll help us understand how can we work to design drugs or mechanisms to target cancer. If you're interested in any of these programs, we do have some specialist master's programs. Thank you very much for listening, and I hope you found this video helpful in your own studies. Bye.